Hello, everyone. Thank you for attending today's webinar entitled Bacterial Viruses as Alternatives to Antibiotics, presented by Dr. Katrina Whiteson, an associate professor at the University of California, Irvine, and the co-director of the UCI Microbiome Center. Her lab studies the human microbiome in health and disease. Before we begin, I just want to cover a few housekeeping items. At the bottom of your screen, you'll find multiple application engagement tools that you can use. All of these tools are resizable and movable, so feel free to move them around to get the most out of your desktop space. Today's presentation will be followed by a live Q&A with our speaker. If you have any questions during the webcast, please submit them through the Q&A engagement tool located at the bottom left of your screen. We will do our best to get to as many of your questions as possible at the conclusion of the presentation, and the rest may be answered later via email. Please know that we do capture all questions. Additional help materials are available in the resources list on the bottom center of your screen. We encourage you to click on any links that you may find useful, including the application note that will be featured in today's presentation, more information about Dr. Whiteson's lab and the work being done with her team, as well as the Agilent BioTek LogPhase 600 microbiology reader. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background that could cause issues. And for the best audio quality, please make sure that your computer speakers or headset is turned on and the volume is up so you can hear our presenter. Additionally, an on-demand version of today's webinar will be sent to you by email in the following days. Again, I want to thank you all for joining us today. And with that, I will turn things over to Dr. Whiteson.
Great, thank you very much. I'm so glad to be here. And I'm excited to tell you about our research in our lab focused on using bacterial viruses, or phages. I'll refer to them as phages in the presentation. And here you can see our team in action. This is a team of students in our group using this Agilent plate reader, which has been really a favorite instrument in our lab this year. So I'm excited to show you some of the data that we've generated and just to tell you some background about how we got there. So I'm at UC Irvine. And here, I'm just going to advance. So here's the instrument. We did write a technical note showing the method that I'll share some of the data from today. And so you're welcome to check that out to get more of the details. And I'll give you more of the background of how we got there really in the talk today. But here you can see the details about the instrument, and our method is really well described in this technical note. So we're at UC Irvine. We're between San Diego and Los Angeles, so we're near the ocean and the mountains. And I've put my email address here in case any of you guys would like to be able to contact me or ask questions later. And I want to start with the acknowledgements. We really have a great team in our lab with undergraduate students, PhD students, and postdocs. And I'm acknowledging many of the people on our team. Here's a picture of us on Catalina Island last fall. We all went for a retreat for the day. And I'm also thanking some of our funders here who have made a lot of this research possible. So bacteriophages are viruses that infect bacteria. So I'm showing you an image here of a bacterial cell. It's an E. coli with a bunch of these phages, which are iconic. In fact, sometimes you even see SARS-CoV-2 represented like one of these phages, although the morphology of SARS-CoV-2 viruses is completely different. So this image has captivated the public eye, I think, really representing what we think a virus looks like. And a lot of phages do look like this. It's now been more than 100 years since phages were discovered. And many people have already been using them as alternatives to bacteria. Even before we had antibiotics, people were using phages as alternatives to antibiotics. Think of it like the enemy of my enemy is my friend. So if you can find a virus that'll kill a bacteria causing an infection, it can be used as a treatment. So as I just wanted to put in context the size of the viruses. And so you can see down here, Zika viruses and then bacteriophages. Here's an iconic bacteriophage of the same style in the image that I showed just before. So they're really tiny, like maybe a quarter of a micron in size on average. And you can see relative to coronaviruses how big they are. And I guess importantly, comparing them to the size of bacterial cells is useful too. Bacterial cells are usually 1 to 3 microns. Great. So as I mentioned, phages were discovered more than 100 years ago. And here's some of the history in the ways that phages have impacted biology and medicine and research. So immediately, some of their early discoverers had this idea that we could use them as a treatment to treat bacterial infections. And there's wonderful stories actually from this era, even before we had antibiotics. For example, Elizabeth Taylor was treated for a staph infection while she was filming the movie Cleopatra in the UK using phages. There's other amazing stories like that from a long time ago. And then more recently, because of the crisis in antibiotic resistance, there's starting to be some amazing stories of modern cases of people who have infections that are not able to be treated with antibiotics. And then we use phages in a compassionate use exemption context, and it ends up curing the infection in some cases. So I want to back up and tell you a little bit more about the life and times of phages just in normal times. So, I mean, in nature, every time there's a bacterial cell, any environment you can think of, the ocean, your chair, etc. There's phages and bacteria everywhere. Every time you find a bacterial cell, there's probably about 10 times as many phages infecting that cell. And the circle of life where the viruses are killing those bacterial cells and releasing nutrients ends up being a really big part of how microbial communities are thriving in nature. So in the ocean, for example, half of the oxygen on our planet is produced by bacteria in the ocean. And every day, phages kill a big fraction of those bacterial cells in the ocean, unleashing nutrients, helping that population of cells turn over. So that's a really important way that phages affect the world. They're also really good at carrying genes and transferring them to other cells. So a phage, of course, has a little package of DNA with instructions for how to copy itself, and it carries other tricks too. So if it's going to help the bacteriophage make more copies of itself, it might carry stress response genes, or antibiotic resistance genes is a really famous case, so that you see phages carrying interesting genes into other cells. And because of that, and in general, they are drivers of bacterial evolution and diversity. So if I were to take a sample from somebody's gut today and again in 3 years, you would find really significant evolution, and the phages would be a really big part of that. So in applications, there's a bunch of great contexts that phages have been used in. They've been used as model systems and tools for molecular biology. A lot of the initial work on the discovery of what DNA was and how it replicates, a lot of that work was done with phages in the middle of the 20th century. They're great models for studying evolution. That's something my lab is doing. They have very fast turnover time, so you can get a lot of generations quickly. So a PhD student on a short timeline of a few years can do a lot of experiments with bacteria and phages. And then, of course, this really exciting context that I've mentioned, phage therapy. So the idea with phage therapy is that you can use the viruses to kill a specific bacteria. Most phages are quite specific in what type of bacteria they can kill. And so as a result, you can have a virus that comes in and targets the infecting bacteria. It doesn't infect human cells, and importantly, it doesn't infect other kinds of bacterial cells. So as opposed to antibiotics, which might decimate your healthy microbiome, phages could hopefully specifically target the infecting bacteria and leave the rest of the microbiome intact. So again, the idea is, the enemy of my enemy is my friend, and you can use viruses to have them on your side to kill the infecting bacteria. Here's an ad from Paris in 1936 of a bacteriophage preparation that was being sold in pharmacies there. So you can see this really is an old idea. And this still happens. There's parts of the world that these kinds of cocktails are still being prepared and sold. For example, in many of the former Soviet republics, like in Tbilisi in Georgia, there's a famous institute where these kinds of phages are still sold. And no fundamentally new antibiotics have been discovered for 20 years. There's a combination of science and politics and pharmacy and economics that have led us to this conundrum, but we really are not in a turbo mode for discovering new antibiotics right now. And every time we do discover a new antibiotic, resistance arises soon thereafter if the antibiotic is out in use in the world. So we really have a big crisis, and the number of deaths that are projected to be associated with antibiotic-resistant infections is really staggering. It's like thought to rival cancer in the coming decades. So the idea with phage therapy is when antibiotics are not there for you, you could use a phage. And there's been a few really famous stories of people who have had success turning to this strategy in the last few years. One of my favorite stories is Stephanie Strathdy at UC San Diego, whose husband, Tom Patterson, had developed a terrible Acinetobacter infection while they were on vacation in Egypt. And she remembered learning about phage therapy when she was an undergrad in Canada. I love telling my undergraduate microbiology classes this story because maybe they'll learn something that will help their family way in the future. And so she spoke with the director of the infectious disease department in her husband's hospital about the idea of using phages, and they started this worldwide hunt to find phages that could help his infection. And in his case, it was a big success, and they've written a book about it. It's a wonderful story. So there's some applications where phage therapy is thought to really have some advantages. So for example, in infections where circulation is poor, so it might be hard to get antibiotics to the spot where you need them. If you were to use a phage, even just getting, potentially very small numbers of phages, just a couple, to the right spot where there are cells where they can replicate, in theory, you increase the dose of the treatment that you need as the phages are replicating in the place where you need them. So that's a really amazing idea that could really make phage therapy work, for example, in chronic wounds or in places where circulation is poor. And then, of course, in cases where the bacteria are resisting antibiotics, they might not be resisting phages. And it could even be that we should be doing combinations of phages and antibiotics together. Sometimes they have synergy. That's actually a big area of research in our lab too. So here's a little bit more about some of these stories of successes with phage therapy. So I told you about Stephanie Strathdy. Here's a link to a video about it where she talks about remembering that she had studied bacteriophages when she was an undergrad. And then I'm showing you some images from the Eliava Institute in Tbilisi, Georgia, where they are using phages. It's a regular thing that people line up whenever they have stomach troubles or other infections, and they buy these cocktails like the one shown in the package that I've got a picture of here on this slide. And in this video, they go through what it's like to be a patient at this institute, and how they find a phage that works for your infection, even if one of the typical ones that in their prepackaged cocktails doesn't work. They'll reach back into the freezer and find you a phage that might help. So it's really like personalized medicine in a really interesting way. And here's a New Yorker article that features actually my postdoc advisor from San Diego, Forrest Rower, talking about this new trend of people turning to bacteriophages when their infections are not treatable with antibiotics. So as I've mentioned, there's a few cases where the phages have some really interesting advantages. So here's a table comparing the use of antibiotics with phages. And, really, I guess a big take-home message from this is that the idea is that the phages will replicate at the site of the infection. And so they might have advantages in places where it's hard to get treatment with antibiotics. This was a really wonderful story written by Sinclair Lewis, Aerosmith, and the whole storyline, they were searching for like really cutting edge science to put into this novel, and he chose the idea of bacteriophages, which became the protagonist in the story's kind of life quest. So Sinclair Lewis had been hearing about the idea of bacteriophages from his friends at the Rockefeller Institute. And it's a great novel. And I thought it was fun to point out that even in 1925, this idea was captivating the public imagination. Okay, so here's a question for you. I wonder if you guys have a way of responding to me, but maybe just give yourself a chance to read it. So there are more bacteriophages on Earth than, and I've got some options here, and I will just go through them. So as far as the numbers here, really the number of phages in the universe is greater than any of these other cases that I mentioned. So there's thought to be 10 to the 31 phages on Earth. And keep in mind, for every bacterial cell, there's 10 phages. It adds up really fast. There's a lot of environments on the earth that are teeming with microbial life. And it's true for really every kind of cell, that every kind of cell has viruses infecting it. And there's just so many bacteria that the diversity of bacteriophages becomes mind-boggling. So yeah, as I mentioned, there just is incredible diversity of viruses, and I hope this is motivating. There's so much to learn. We really are in the early stages of, we've barely scratched the tip of the iceberg in terms of understanding viral diversity. In fact, one of the most common phages in the world was only very recently discovered. This is a gut phage that's known as Cras phage. And they're using this nice depiction with the Matryoshka dolls about, the microbes in the gut are infected by viruses themselves. And this virus is present in at least half of people and very abundant. And we had no idea it even existed until a few years ago. So here's some images of the different types of morphologies of phages. So the famous one that I showed you earlier in the talk, that T4 bacteriophage style, is a myovirus like this one, depicted up in the upper left. But that's the iconic image of a phage, but they can take shape in many different types of forms. And I wanted to share the diversity of how some of them look. So there's a lot of really interesting morphologies that phages can take. And in my lab, I think these top three, the myo, cypho, and podo, are probably the most common type that we end up working with. And I hear a lot about filamentous phages, like these innoviruses with the big long strands. So I just want to make sure I'm not biasing myself towards the iconic image, because there's a lot of diversity out there. And that's actually been motivating in our group. We recently wrote a perspective about how we should be making sure we include all of the unknowns in our data sets when we publish papers. So since DNA sequencing has become cheaper in the last 15 or 20 years, it's been such a fun time to be a microbiologist, because we're constantly generating these huge sequencing data sets that in many cases are shotgun and unbiased and capturing every microbe in the system. But many people will then run the data, compare their sequencing data to a database. So they're biased towards what other scientists have discovered before them. So I challenge you, every time you read a paper that has metagenomics data in it, look and see if the authors report how many unknowns were in their data set and did not end up becoming part of the main analysis that they're showing. Sometimes people are only analyzing 5% of their data because most of it is just totally undiscovered. So that's something we're always trying to do, is report the number of unknowns in our data sets. And those unknowns are often phages. So I'm hoping this feels motivating, that there's just a lot to learn, and you guys who have labs out there are in a position to make really big discoveries, because there's so much to learn still. So it can feel daunting, like, oh my gosh, how are we going to be able to make progress in this field where there's still so much that's undiscovered? But I have some really great collaborators, and so I've had a lot of opportunities to get to work together with people, which is making it feel more possible. I'm sharing here a recent collaboration with UC San Diego, where we're working on Stenotrophomonas phages, which cause infections in people who have immunocompromised status, including cancer patients and people with cystic fibrosis. And down at the IPATH, the center in San Diego, they get just a staggering number of requests for help with phage therapy. And so you can see, more than 1,000 in the time period listed here, the majority from the United States. And at this time, there's not really like the bandwidth or the right phages or even the medical infrastructure to help many of them, but we're slowly making progress on figuring out how to deploy the phages in a timely way and in an effective way. So in our lab, phage hunting starts with the help of our team. We call ourselves the Orange County Phage Team. So this elite team will get sewage samples and incubate with a bacteria of interest. So for example, here's our Enterococcus team. Enterococcus is one of the escaped pathogens. And then what you see here, this is a plate that has a white bacterial lawn. And whenever you see a circular clearance zone, which we call a plaque, that's evidence of a phage infecting the bacteria. So the phage is eating the bacteria and clearing away that white lawn. So we'll end up with new phages. And we've done this for a bunch of different targets. Here's our Stenotripomonas team that I mentioned. So here's the posters that 6 of the students presented at our symposium this year. Many of them running the Agilent plate reader, actually. So they're really big fans of the plate reader. And so you can see the kinds of projects that people are doing, varying the temperature while we're doing our phage hunting, and characterizing the genomes as we sequence the phages that we discover. So anyway, it's been really fun to have this vibrant group of students helping us with our research. So as I mentioned, we'll start from raw sewage, we'll incubate with a pathogen of interest, and then we'll look for plaques and pick one of them and propagate them. And this is not the glamorous part. It can sometimes take many rounds of purification to get an isolated individual phage. But that's the goal, so that we really know which phage we're working with. And then we'll filter, so we've got just the phages by themselves in the liquid that we're working with. And actually, during the pandemic, the fact that we had all this sewage in the lab had a really big impact on our lab's research, because we were able to sequence the sewage in the search for other pathogens, especially SARS-CoV-2. So we've actually now written a bunch of papers about the wastewater sequencing. And we did several projects in collaboration with the Southern California Coastal Water Research Project, where we got samples from 8 different wastewater treatment plants. And from those samples, we actually represent 16 million people. And here's an example of one of the papers that we published describing the different kinds of microbes that we found in the RNA from the sewage. There were so many interesting and surprising things about this study. For example, we had to pasteurize the sewage, so incubating at 70 degrees C for an hour for safety reasons, of course. And then, our goal was to use transcriptomics, because SARS-CoV-2 is an RNA virus. So imagine if someone told you that you had to cook your RNA before you could work on your RNA-seq project, which really didn't seem like it was going to work very well, but it actually worked really well. We made some really cool discoveries and got some great data from these projects. And I want to highlight postdoc Dr. Jason Rothman, who led all this work. And then, I know you guys can't respond, so I'm just going to ask this question. Which virus do you think is the most abundant in wastewater? And when I ask this question, a lot of people will guess, phages, maybe E. coli phages, which is a really good guess, especially since all I can talk about is phages. Or sometimes they'll guess a human virus like influenza or something like that. And we did capture that, especially norovirus was in almost every sample, even while we were all quarantining. But interestingly, the most abundant virus in our wastewater samples were plant viruses, especially this tomato virus. It's a Tobama virus. And Jason just published a paper about these Tobama viruses, actually. And so anyway, that surprised us. We knew that pepper mild mottled virus, PMMOV, was going to be abundant. That's famously the most abundant virus in fecal samples too. But this was a surprise. So we're really we're really trying to figure out what's going on here and why we have so many tomato viruses in the sewage. Okay, so back to my main topic at hand. We're aiming to harvest phages out of the wastewater. And so, as I mentioned, we will incubate sewage with a pathogen of interest. We'll plate them out, and then we'll look for these clearance zones, which represent the phages growing on the white bacterial lawn. So we make these plaque assays. Plaques are the clear zones that are formed in the lawn of bacteria due to the phage lysing or eating and killing the bacteria. So ideally, each plaque represents an individual phage. So if you spread them out enough, you can really get at individual phages using this approach. And it's a really old and famous approach. It's amazing how much science we've done with this in many ways, simple method. Here's an example. We had the doctors down at UC San Diego contact us asking if we had any Stenotripomonas phages to help a cancer patient. And some of the students in the lab immediately in one of our samples found actually very abundant phages. So you can see on this plate, there's so much phage activity that the the plaques are kind of blending together. There's a lot of clearance on this plate. So that was kind of the start of our Steno project, actually. But today I'm going to tell you some more about our Enterococcus project and land with some of the data that we've been generating with the plate reader. So Enterococcus can be a friend or a foe. It's one of the escape pathogens, and about 30% of Enterococcus infections are vancomycin resistant. It's also a very common gut bug. It's usually about 1% of an average healthy person's gut microbiome, and they've been associated with the guts of all of these creatures back to the split between sponges and nematodes. So it's a very ancient association. But it's an important pathogen, and interestingly, when people take antibiotics for other reasons, sometimes one of the consequences is an Enterococcus infection, because Enterococcus is so good at resisting antibiotics in stressful conditions. So yeah, here's an example of a study we did at the Children's Hospital in Orange County, where we were just looking at the microbial composition of babies who either had good outcomes. All of these were preterm infants, and some of them were fine, and others developed sepsis or necrotizing enterocolitis. And we were looking to see if there were specific microbial associations with those difficult outcomes. But really what we found is just that all of the babies were getting a lot of antibiotics, and many of them had really a lot of Enterococcus in their gut microbiomes, which is not what you're hoping for. You're hoping for high amounts of bifidobacteria in baby gut microbiomes, and you can see that's really not happening here. So that was motivating. You see this Enterococcus overgrowth. So we were looking to see if we could have a more targeted approach using phages to target these kinds of infections. Now, if you look out at the number of sequenced phages in the world, you'll find that there's actually very few Enterococcus phages. So we really needed more. So we sent our phage hunting team out. And as you know, we've been able to isolate phages with this approach. Here's some of our Enterococcus phages. Here's a collection of 20 that one of our phage hunting teams gathered. And you can see there's several types that we've gathered. So we've got myoviruses that have really big genomes, like 150 kilobases, and their plaques tend to be a little bit hazy. And then we've got cyphoviruses with smaller genomes that make these much more clear plaques. And then podoviruses with even smaller genomes, and you can see their plaques really are quite clear. So I'm going to focus a little bit on these myoviruses, which have turned out to be very interesting. They have a lytic lifestyle, so no evidence of integration into the host. They have a broad host range, so they can infect a lot of different kinds of Enterococcus. And they typically encode about 200 genes, and here's an electron microscopy image of them, so you can see that kind of myovirus typical morphology. When we take the sequences and compare them against all of the data in the sequence read archive using, this is a really cool approach developed by Rob Edwards called Search the SRA, and you can see these phages are showing up in data sets from all over the world. They fall into two main groups. So you can see on their genomes that there's two clusters of shared genes that are quite distinct from each other, even within these myoviruses. And interestingly, one of the clusters is mostly able to infect Enterococcus faecalis and not so much Enterococcus faecium. And the other one is having quite broad host range and can infect both E. faecalis and E. faecium. And yeah, we've had some interesting observations about the biology of these different groups of phages. For example, the ones with narrower host range encode fewer tRNAs. And then here's some of our other phages, the cyphoviruses and the podovirus, just one podovirus that we've discovered. And they, for the most part, are a little bit more sporadically infecting the strains that we have. So I want to call out what I mean by host range. So host range describes the breadth of organisms that a parasite is capable of infecting. So I've just shown you what I'm calling host range data for our viruses infecting bacteria. And this term applies if you're talking about, influenza capacity to infect different organisms. So here's different influenza types where some can infect people and specific animals. So, for example, you could say that the IAV strain here has a broader host range and can infect a much larger number of different types of animals, for example, than IBV, which is restricted to infecting a smaller set of animals. So sometimes we want to be able to expand the host range of one of our phages to be able to infect a new pathogen. And so we've resurrected this century-old strategy that they've employed in some of those clinics in the former Soviet republics, like the Elyava Institute in Tbilisi, Georgia. It's called the Apelman's Protocol. And so essentially, the idea here is that we will take a really recalcitrant strain that none of our phages are able to infect, and we'll mix it together with other hosts that the phages are easily able to infect. And we do this for many rounds with the hope that mutants will arise through their infection of the more easily infected host and then be able to help us take down the really recalcitrant host. So I like to make this analogy for the students that, imagine you're trying to convince a toddler to eat some broccoli. Well, maybe you could put some broccoli and some mac and cheese on the plate, and they'll start out with the mac and cheese, but then when that runs out, maybe they'll turn to the broccoli, and their appetite is going at that stage. And that's exactly what we're hoping is going to happen in these phage communities, that we're going to get the infections going on the more easily infected bacteria, and then mutants will be generated, and hopefully some of them will reach out and be able to infect the broccoli, the recalcitrant strain. So I'm going to show you a comparison of two different types of cocktails we've built recently, where we've compared taking a bunch of our sister phages. So these are the myoviruses I've been talking about. So we put 3 of them together against 3 different bacterial hosts. So DP05, that's our mac and cheese. It's really easy to infect. Whereas DP15, that's like our broccoli, the difficult to infect strain. You'll see lysis is represented in red here with a plus sign. So all of our phages can infect DP05, whereas for DP15, none of our phages started out at being able to infect. And we did this evolution Apelman's experiment with 2 different types of cocktails. One was 3 different myoviruses, so closely related sister phages. And then we also did a similar evolution experiment, but with cousin phages. So we put myo and some cyphoviruses together that were more distantly related. And so we tried out this evolution experiment. It went on for 20 different cycles. And so in plot A here, what I'm showing you is the dilution factor. So like the amount of phage that was needed to be able to take out the hosts. And so more dilution. So up at the top of the plot, that's a 1 to 10,000 dilution. That means there are very, very few phages being used. So if you see this very dilute phage taking out the bacterial community, that tells you the phage is super potent. So these 3 top bars with the where you see that 1 to 10,000 is enough to get a signal, that tells you the phage was really potent. And it didn't start out that way. So even just after a couple of cycles of this evolution, you see that the phages became quite capable of taking down the strains. It was slower for the DP15 on its own, but you do start to see activity after a few rounds. And by the end of round 20, for both the sister and the cousin cocktail context, we do see that the recalcitrant strain was able to be taken out. And the same data is shown in a heat map form over here on part B. And you can see that the dilute phage is shown with a darkest red color. And after a couple rounds, you see that the bacteria are able to be taken down with the evolved phages. So, and here's a more direct way of showing you this. Here I'm taking the time point 20 final evolved cocktail and showing you their capacity to take out the different bacterial hosts. And DP15 is our broccoli here on the bottom. And you can see really there was no evidence of clearance in the ancestral starting point. But with the evolved phages, both the sisters and the cocktails, you see these clearance zones. They're a little fainter on DP15 than on the other hosts, but there's still definite evidence of activity. So the pinnacle here of my talk, I wanted to share some of the data that we've been generating with the Agilent plate reader. And so what we did was we wanted to learn about the host range of our evolved phages that I just told you about. And so our team wrote this technical note showing how we've been doing the experiment. So you've got lots more details you can take a look at. Here's our protocol, and this is part of the technical note actually. So please feel free to follow the details there. But basically we take our bacterial strains. And so because we've got so much capacity in that plate reader, we have 4 different 96 well plates. So we dedicate one of them to the bacterial control. So we can test 90 strains at a time. So we got a bunch of different clinical isolates of Enterococcus from our main colleague in San Diego, David Pride, sent us all these Enterococcus strains. We put them all on the plate together. Then we have one of the 4 plates as the bacterial control, and then we do triplicates of our phages in the other 3 plates. So in just a couple days, we have triplicate data for a phage against, 90 or so different hosts. It's been so fun to get the data back so quickly. And so I'm excited to show you some of what happened. The ancestral phages, so the starting point phages, are shown on the left of the plot and indicated by this green bar in the top left. And then the evolved, these are some of the isolates. We took individual phage isolates from our evolved cocktails. They're shown with an orange bar up on the top right of the plot. And the darker the color, the more complete the suppression of bacterial growth is. So complete suppression is where our black bacterial control is growing great, but the phage is completely knocking out growth. And we've categorized them. Some of them, it's a more of an eventual suppression, so it doesn't happen as quickly. Others eventually resist, so the suppression doesn't last for the full 72 hours of our plaque assay. And we're going out really long with the plate reader, which has been great. An average clinical microlab test occurs at 18 hours, but we want to know over the longer term how long the phages are able to continue suppressing bacterial growth. So then we have our next categories where they're mostly resistant. And then if we don't see any impact of the phage at all, then that's just resistant. So the darker the color, the more complete their suppression. So you'll notice our ancestral phages were unable to take out many of the bacteria that are still showing up in light colors, especially in the top right of the plot. Our evolved isolates are much more able to take out many more of these Enterococcus faecalis strains. And here's actually a quantification where you can see that the percentage of strains that's suppressed has really increased for the evolved isolates. Just to give you a sense of what the data looks like, here's what the bacterial control plates look like. So each isolate kind of has its own growth pattern, and it's really cool that we know this about the strains now. If we were just doing spot titers on agar plates, we wouldn't have this kind of information. Here you can see what it looks like when we combine the bacterial control, and then now I'm showing you the phage growth patterns. So all of this is triplicate data for a bunch of the different phages that we've been working with. And this data appears in the technical notes, so feel free to have a closer look. But I really love getting to see these patterns. Having the kinetic data is really valuable to us. It changes what we learn about the interaction between the phages and the host really significantly in comparison to just seeing those plaques on the agar plates. So here's just a really fresh right off the press new plot where I've got some additional data, and you can see I've got now the cousin and sister cocktails in the ancestral column, so we can see what the starting point is for the cocktails. And another important thing I'll point out about this heat map, which I just showed you a second ago, but in purple are Enterococcus faecalis strains, and in green are Enterococcus faecium strains. All of the hosts that we did our experiment with started out as Enterococcus faecalis hosts. But we were curious if our evolution expanded them to be able to infect Enterococcus faecium. So far, that does not seem to be the case. So while our host range is very much broadened, especially for this top cluster of purple Enterococcus faecalis strains, so far the evolution experiments are not expanding us into the epitheliums. And so we're constantly collecting this data and plan to publish a paper about this project soon. So stay tuned, or if you have suggestions, we're really listening. It's a really fun project in our lab right now. And here's our team again with the instrument. And so Andrew led the technical note, he's here. And Alicia is a PhD student in the lab who actually was the person who discovered that this instrument exists and convinced me that we should go for it. And so in conclusion, the BioTek plate reader has just really been great at helping us successfully identify lytic phage activity in our kinetic measurements. And as a result, we have a higher throughput approach for identifying effective phages and for characterizing the new properties that emerge in our evolution experiments. And so, yeah, the use of multi-phage or phage antibiotic cocktails can provide valuable alternatives to antibiotics. And in fact, I didn't show them here, but we've also been using this plate reader for our antibiotic and phage synergy assays, which also occur in the 96-well plate format. So with that, I will make final acknowledgement for the team that enabled all this work. I especially want to thank David Pride at UCSD, our collaborator on both our Enterococcus and Trichomonas projects, and just our great team of students who have so much enthusiasm and have made all this happen. So thank you very much, and I would be happy to take some questions.
Awesome. Thank you so much, Katrina, for a great presentation. If you haven't had a chance to submit a question using the Q&A, you can just type it right into that chat box, and we'll get to as many as we have time for. So it looks like we do have a few in here now. So the first one is, do you think that phages will become an alternative to antibiotics in the future?
That is a great question. You know, I know they have the potential to. So it's something that all of us in the phage biology community are thinking about a lot right now. I was just at the Evergreen Phage meeting in Olympia, Washington last week, where there were 200 phage biologists, and this is something we're all thinking about. Everyone has their own strategies for trying to make it happen. Phage Australia has a bank of 500 phages, and Phage UK similarly probably has 500 or 1,000 phages. And here in San Diego, or you know, I'm in Irvine, same thing. We've all got these banks that we're building up, and we're all willing to do the work to help people get compassionate use exemptions on a case-by-case basis if they present with a infection that we think we can help. But right now, it's still very boutique. It's not standard. So it's a lot of work to help someone get phage therapy access right now. So whether that becomes easier is a great question. You know, the FDA has made this pathway work for us to be able to use it in a compassionate use exemption context. And I personally am very curious how it's going to go in the next few years. But I do think that we will continue. The phage biology community wants to help and make this happen. And, you know, we need resources to turn it into something more streamlined. And, you know, as a testament to the longevity of these approaches, in Tbilisi, Georgia, at the Eliava Institute, they've been using phages for 100 years. So that helps give us a template for a place where it works.
Let's see. Ooh, quite a few are coming in. How are phages stored and prepared so that they remain safe and in good shape?
Great question. We actually have, that's a kind of common project for new students in our labs, to take one of our phages and try storing it in a bunch of different conditions and then going back and checking it every few months to see if it's still alive. It's very individual for each phage, so it's been really fascinating to figure that out. And, you know, you hear stories like at the phage meeting, you hear stories of like, oh, we inherited an old fridge from a neighboring lab that had staph phages that are 20 years old and they were still active. So that might not be true for every phage, but it often is. And so how do we personally do it? We actually store things both at -80 and in the 4 degrees. So when we make a stock, we make a bunch of aliquots. And in our tests, we've been comparing the use of glycerol and DMSO. And then really a very important strategy is to store the cells together with the phages together so that the phages can be protected inside the cells. So we will infect a new fast-growing group of cells with phages, and when they've had a chance to infect, then we'll put that whole mix into the -80 so that you can resurrect the whole culture, and hopefully the phages are protected.
Let's see, here's one. How many different bacteriophages could be used today?
How many different? I mean, so really it's endless. There's essentially infinite diversity of phages. We haven't even come close to saturating the discovery of the phages. You know, every time we discover a new phage, not every time, but many times that we discover a new phage, it has no sequence homology to any other discovered phage. And so, yeah, how many? I mean, the banks typically have a few hundred phages. I would say, actually, I just saw a presentation from a company, Locust, and I think they said they had 6,000 phages. So, and that's after a couple years of intense hunting. So if you get a team of people together and you do a combination of borrowing and hunting, you know, you could get yourself up to hundreds or thousands of phages pretty quickly, I would say. Of course, you need a specific phage for every infection, so that doesn't mean we've covered all the pathogens, but I think we're getting there. Great. So let's see. Great, we've got a lot of questions.
Coming in. Yeah. We'll just keep plowing through. Does that sound good?
Yeah, please do. Sounds good.
Okay. All right. So here's one. What is the mechanism of the development of phage resistance, and is it due to the changes in the target of the receptor binding domain?
Yeah, wonderful question. So how does phage resistance arise? So, I mean, every time phages and bacteria are co-evolving together, there's this arms race, and the phages mount strategies to infect better, and the bacteria mount strategies to resist better. In our Enterococcus experiments, we've sequenced both sides of that coevolution a lot of times. And most typically, we see mutations in the tail fiber proteins arising on the phage side. There's a few other genes and intergenic regions where we typically and very commonly see mutations arise too. And then on the bacterial side, it's interesting. It's almost always in the exopolysaccharide biosynthesis clusters that we're seeing mutations accumulate. So I think that's more like the surface sugars obfuscating a receptor. I'm not sure what the receptor is unless the tail fibers are just essentially engaging with those exopolysaccharides sugars. That's where the mutations are accumulating. I love coevolution as an approach for learning about that. And then I would just say that we do sometimes see mutations in other genes, like, you know, ribosome related genes, which could actually also be important antibiotic targets. And so that has given us enthusiasm for synergy with antibiotics as well.
Is bacteriophage currently in use in clinical settings? I heard that it is being used in Walter Reed Hospital.
Yeah, so around the world right now, there are many different hospitals that are working with phage biologists to use phages in a clinical setting. They are not approved in a general way. It's a case-by-case compassionate use exemption context. So if a patient has a recalcitrant infection and wants to try to go for phage therapy, then there's a series of steps that have to be taken. And if you look at the IPATH website, there's a nice fact that lays out the chronology of those steps. But essentially, the doctor has to apply for an exemption from the FDA to be allowed to use this experimental therapy. So it's really case by case, and it's only in a compassionate use exemption context. But it's happening. I mean, I'm part of a paper that we just published that had 12 cases in it, and our Enterococcus phages were used in that paper. So,
and just for the person that asked that question, you can link to the IPATH website in the resources list. So it's right there for you. Let's see, we got another one here. Is phage therapy being utilized for MRSA infections?
For MRSA staph infections, yes.
Yeah,
it's interesting. We and some of our colleagues have tried to isolate staph phages, and we have a few. It's been a little bit of a harder bug to hunt phages for, it, you know, just as a word on the street. I've been hearing that, but yes, there are examples of phages that can infect MRSA strains, and it's a really important context. We just wrote a grant across a bunch of the University of California campuses with the goal of developing phage therapy for prosthetic joint devices, which often get staph infections, and so the focus of that project was all on staph phages. So yes, phage therapy, I would say, is being developed for MRSA, and I'm sure there are sporadic cases where it's been used, but it's not like a general treatment yet.
Gotcha. That's exciting. Let's see, a couple more. Is there the possibility of using phages to help cure or treat cellular diseases in mammalian cells?
I mean, so phages, bacterial viruses, bacteriophages are very specific to infecting bacterial cells. So, so phages are not being used in any context that's intended to infect a eukaryotic cell. There are treatments that involve viruses that do target eukaryotic cells. For example, there are cancer treatments being developed that involve viruses that are able to infect cancer cells. Or viruses that invoke an immune response that helps with other disease states like cancer. But yeah, phage therapy is really very specific to bacteria.
Let's see. Are there any examples when a bacteria developed resistance to a phage?
Yes, so exactly. There are these arms races where bacteria resist the phage, and phage mount a better infection strategy or a different infection strategy. So those arms races are happening all the time. I mean, in your own gut with very dense bacterial colonization, that's happening a million times over right now. So that's a constant occurrence. And we see that the bacteria sometimes evolve resistance to our phages. I don't see that as a different problem than antibiotic resistance. I mean, if you give antibiotics, then bacteria will develop resistance strategies against the antibiotics as well. So ideally, the phages are like another trick up our sleeve, and maybe the synergy between antibiotics and phages will be an orthogonal attack that is even harder for bacteria to mount a resistance to. We've certainly seen that with cocktails. We just published a paper about Enterococcus cocktails, actually, where we showed that if you add together 3 different kinds of phages, the resistance is essentially completely suppressed for the entire 72 hours of our assay. Of course, you know, every context in an infection will be a little different, but yes, bacteria can mount resistances against phage, and that's, to me, that's not a concern. That's not a different problem than we have with antibiotics, and our goal is to bring the bacterial load down a bit so that the immune system can help. And that's what antibiotics do too. You know, you want to reduce the bacterial load so that you can clear the infection. And so I think bacteriophages could be just a great strategy to help make that happen. Cool.
Let's see. What do you see as some of the barriers to the clinical development of phages?
Well, I mean, there's a lot of different kinds of barriers that I've been bringing up for the clinical development. I mean, first of all, it's an unusual situation. It's much more like a personalized medicine where for each strain of bacteria, you might need a different phage. So, you know, for example, if someone told me they were going to design a clinical trial to test the efficacy of phages and they wanted to use the same phage for everybody in the study, but then people come in with totally different kinds of infections and maybe the phage only infects one kind of bacteria, that would be a very ineffective study, right? I mean, that just doesn't work. So we have to come up with new models for how to set up our studies in order to learn about the safety and efficacy. And it's tricky because each phage is different. So each phage could have different genes or different storage conditions. So it's going to be a challenge to make that work. Some of my favorite strategies right now include, at Phage Australia right now, for example, they're doing a clinical trial where they're just standardizing a lot of the steps in the phage therapy treatment, but they're allowing the individual phage to be used. So in their study, you know, you come in, they'll go find a phage that works for your infection. They'll prepare it in as similar a way as possible, but they're not trying to use only one phage across the whole study. They're allowing people to choose the best phage for that particular infection. And then they'll gather data on, how the whole thing goes for people across the use of all those different phages. So we're going to have to be a little bit creative about how we set up our clinical trials to learn about it. So that's a challenge, I think. And so is, we right now we don't have general manufacturing of GMP level phages happening. There's a few labs in the world that can do it. And it's hard to get, pharmaceutical companies are not going to invest in this type of technology because it's they want more chronic medicines that can be used through a longer period of time. That's why antibiotics are harder to invest in. So phages have a lot of the same challenges as antibiotics for getting investment in research to happen.
All right, so I think we got time for a couple more here. Let's see, how can you determine the amount of phage to a specific concentration of bacteria needed to kill it completely? Has this been tested on an animal model infected with bacteria?
So first of all, how do we figure out the concentrations? Or the multiplicity of infection? That's a great question. So we count the titer of our phages against the strain that we propagate on. And so it's an interesting number that you catch. Like there's a lot of different ways you can count things. You can try to look at them and count them. So we could like fluorescently label them and look at them. That was a really popular strategy about 10 years ago, but we've discovered that there's a lot of artifacts and sometimes you're counting cellular debris rather than phages. And so we've kind of moved away from counting phages, at least in my lab, using fluorescent microscopy. You could use flow cytometry. I love that idea. That's actually something we're trying to develop, but it's not a go-to method yet. It takes some troubleshooting. So our favorite way of enumerating the phages in our lab right now is with the classic plaque assay. So we just make a dilution series of our phages. We add the bacteria from the phage that we propagated on, and then we count the plaques that emerge, and then we learn how concentrated the phages are. So we, typically we want to get them up to like 10 to the 8th or 10 to the 9th per mil. And then we use that to make a ratio with the number of cells. And so for each of our experiments, we target a particular multiplicity of infection. And sometimes we try our experiments at like a range of different multiplicities of infection. That becomes a challenge when you want to work with 90 strains at a time. So you'll notice I was not giving you that kind of information for our higher throughput assay, which was enabled by the Agilent plate reader, where we could do triplicate phages against 90 different strains at a time. So we know that we're using the same amount of phage because we've done the titer count against the propagation host. And so I'm thinking of it in a practical way that, you wouldn't exactly know what the multiplicity of infection is in an infection context either, because it's going to be dictated by local concentrations. And then every time a phage encounters a cell, the number of new phages that replicate and burst out in every cycle will, within a few minutes, completely change your multiplicity of infection. So I guess in my mind, what we're trying to standardize is the number of phages that we put in at the beginning of the infection, so that we can compare in a very apples to apples way across those 90 strains. We added these phages and here's how it looked,
Yeah. All right. Let's see. Let's do one more here. How do you select phages against any particular bacterial strain?
Well, we start with a hunt, like I mentioned. So if we get a new, this happens to us. We get a call from a clinic. They have a strain that is recalcitrant to antibiotics that they might want to try to use phage therapy for, and they send us the strain. If we already have phages in our freezer, we'll start with those. And actually, we've had some really interesting experiences that way. So for many of our projects, we kind of pick a few poster children phages like models. So in our Steno project, we have 3 or 4 phages, which are our most popular go-to phages that are the best studied. So we start with those. Sometimes none of those infect. So then we'll kind of reach deeper into the freezer and go get some of our, other similar phages. Sometimes they have 95% sequence identity with the phages that we've already tested. So we're not sure we're really going to see much of a difference. And sometimes those phages, which seem so similar, now have the capacity to infect the pathogen, which is so interesting because phage infectivity can turn on a dime. Just a few mutations and the whole host range can change quite dramatically. So that's a good reminder of the importance of the microdiversity in the host range. So if nothing in our freezer works, then we'll start looking in some of our environmental samples. We collaborate with local wastewater treatment plants, and as I mentioned, with the Southern California Coastal Water Research Project, so we can get fresh sewage samples. Some of my favorite sewage samples to hunt with are actually ones that have been sitting in our cold room for a little while already, because that means that if there's a phage in there that infects the pathogen, it's pretty sturdy. It's been sitting in the cold room and it's not so delicate. So it's going to be an easy one to store and work with. So we start with the ones we've already isolated and characterized. And then if we don't find one, then we turn to environmental samples to hunt. And you can go with pond water, horse manure, there's phages in so many different substances. So I've heard of people using really interesting samples to hunt.
Oh, that's awesome. Nice. Well, unfortunately, that is all the time that we have today. On behalf of Agilent Cell Analysis, I just want to thank everyone that joined us today, and you, Dr. Whiteson, for your great talk, and that was a great Q&A. If we didn't get to your question, we will be sure to follow up with you via email. And if you would like more information on Agilent BioTek's products and services, including an archive of our previous webinars, please visit us at www.agilent.com. We hope that you enjoyed today's presentation and have a great rest of your day. Thank you.